Okay, get this. Imagine not just seeing where RNA is hanging out inside a cell, but actually grabbing those RNA molecules and moving them around, like tiny molecular puppeteers,
Yeah, it sounds like science fiction almost.
right? But this brand new research has, well, kind of cracked the code on how to do exactly that.
Yeah.
It turns our static view of RNA into something much more dynamic, like a cellular ballet.
It's fascinating because for years we've been mapping RNA locations, spatial transcriptomics.
Yeah.
And we know these locations are super important. Think about specific RNAs gathering at nerve endings to strengthen connections or how they guide embryo development.
Or those stress granules people talk about.
Exactly. Those little RNA and protein hubs cells form under stress. We know where things are, mostly.
Right? We've had tools like MS2 tags, molecular beacons, ways to see the action.
But they were mostly observational, right? Like watching a city from a helicopter.
And often you had to genetically modify the cells, which, might change things.
Precisely. So the big question has been, if RNA location is so vital, what happens if we actually change that location? Can we learn more that way?
That's the million-dollar question.
And this new paper in Nature introduces a tool to do just that. It's called CRISPR-mediated transcriptome organization, or CRISPR-TO for short.
CRISPR-Co.
So our goal in this deep dive is to really understand how this technology lets us basically take control of RNA's address within the cell.
And why that's such a potential game changer for understanding how life works at a fundamental level.
Exactly. And we're using the paper itself as our guide. Programmable control of spatial transcriptome in live cells and neurons, just out last month.
Great. So where do we start? Background.
Yeah, just a little context.
Yeah.
It's crucial to remember that the positioning of messenger RNA, mRNA, inside a cell isn't random at all. The cell puts a lot of effort into this organization.
It's like a really organized factory floor, maybe? Not just making the right product, the RNA, but getting it exactly where it's needed for the next step.
That's a great analogy. And while spatial transcriptomics gave us these incredible maps of the factory floor, our ability to physically move those products around, that's been, well, surprisingly limited.
Right. Which makes it hard to prove cause and effect, doesn't it? Does putting the RNA there actually cause something to happen?
Exactly. You might see an RNA at a synapse that's getting stronger.
Yeah.
But you couldn't easily just, force it to go there and see if that RNA was truly driving the strengthening. You're stuck with correlation, mostly.
Okay, so enter CRISPR-To. How does it leverage CRISPR, which most people know for gene editing?
Well, it cleverly repurposes the core idea.
Yeah.
CRISPR-Cas systems use a guide RNA to tell an enzyme, like Cas9, exactly where to go in the DNA, programmable targeting.
Got it. So how does CRISPR-To adapt that for RNA? Let's get into Figure 1, the blueprint.
Okay, so CRISPR-To has a few key parts. First, there's a special version of Cas13 that's a Cas enzyme that naturally targets RNA, not DNA.
Okay.
But this version is catalytically dead, dCas13, meaning it can bind tightly to RNA, but it won't cut it. Ah, so it's like a programmable sticky note for RNA.
Exactly, a programmable anchor, and the programmable part comes from the guide RNA, the gRNA.
Same idea as in gene editing.
Pretty much. Researchers designed this short gRNA sequence to perfectly match the specific mRNA they want to grab onto, often targeting the 3' UTR, that tail end of the mRNA.
And that guide RNA steers the dCas13 right to the target.
Precisely. So now you have dCas13 reliably stuck to your RNA of interest.
Okay, step 1. Grab the RNA, but how do you actually move it? This is where the magic happens.
This is a really innovative part, yeah. They attached motor proteins to the dCas13 complex.
The cell's own little transport machines.
The very same. These proteins naturally haul cargo around the cell. The researchers basically hitched these motors onto their dCas13. They used a clever linking system involving something called a SUNTAG and an adapter protein. Think of it like molecular Velcro.
Okay, hold on. So the dCas13, guided by the gRNA, latches onto the RNA.
And then this motor protein that's attached just starts walking, pulling the RNA along with it, like a microscopic tow truck.
That's a fantastic way to picture it, and the paper shows just how versatile this is. They could send RNAs to all sorts of places.
Like where?
The outer membrane of mitochondria, the powerhouses. P-bodies, those RNA recycling centers, the tips of neurites and nerve cells, stress granules, telomeres, even nuclear stress bodies, a whole range of destinations.
And programmability is key, right? Choose the gRNA for the RNA target, choose the motor for the destination.
Exactly. You want it at the mitochondria. Use a motor or signal known to go there, need to travel down a neurite. Use a kinesin motor that walks along microtubules. It's modular.
That's really powerful. But getting it to work must have taken some tinkering.
Oh, definitely. The paper details some clever engineering. They use this dual-color reporter system, red and green fluorescent proteins, to visually check how well different dCas13 versions were binding RNA. It helped them optimize the system. That's in the extended data figures.
Did they run into problems, like proteins clumping up? That seems common.
They did, yeah. One of the extended data figures shows that an early version tagged with a blue fluorescent protein tended to aggregate.
Uh-oh.
But they solved it pretty simply by just switching to a different tag, EGFP, which behaved better. Sometimes it's the small tweaks, you know?
Good reminder. Okay, so they engineered it. How did they prove they could actually direct RNA somewhere specific on command? You mentioned an ABA switch.
Right, so to really test the controlled movement, they used a reporter mRNA loaded with binding sites for dCas13.
Okay.
And they targeted this to the mitochondrial membrane, but they used a system that's inducible with a small molecule, ABA.
Abscisic acid.
Yeah, so the idea is the CRISPR-TO machinery only assembles and starts moving the RNA when ABA is present. Add ABA, the RNA goes to the mitochondria. Remove ABA, it stalks.
And did it work?
It did. They clearly showed this ABA-dependent recruitment. The RNA only piled up at the mitochondria when they added the ABA. It gives you this fantastic on-off switch, temporal control.
Super neat.
Yeah.
Okay, so Figure 1 sets up the tool. Figure 2 is about applying it more broadly to the cell's own RNAs.
Exactly. Figure 2 is where they show this isn't just for artificial reporter constructs. They successfully targeted endogenous mRNAs, the cell's natural ones.
Like which ones?
Things like ACTB, β-actin, SGHC, SDHD, NDUFS2, these are all normal cellular RNAs. They move them to the mitochondrial membrane in HeLa cells.
HeLa cells. Okay, a standard lab workhorse.
Right. And showing it works on endogenous RNA is a critical proof of principle. It's not just some quirk of the reporter system.
Did they try other cell types too?
They did. They showed it worked in HEK293 T cells and Neuro2A cells. That's a neuron-like cell line. They targeted GAPDH and Yatch and ECA-B in those, so it seems broadly applicable.
And they made stable cell lines. That sounds important for longer studies.
Absolutely. Integrating the CRISPR-TO components into the cell's genome means you don't have to keep reintroducing them. You can have stable, long-term, controllable RNA relocalization. Big advantage for studying chronic effects.
Okay, and Figure 2 wasn't just mitochondria, right? P-bodies too.
Yep, they targeted GAPDH mRNA to P-bodies using a different adapter protein.
Why P-bodies? What's the question there?
Well, P-bodies are thought to be involved in RNA decay, right? So by forcing GAPDH mRNA in two bodies, they could directly ask, does being in a P-body actually cause this specific RNA to get degraded more quickly under certain conditions?
Directly testing the function of the P-body for that specific RNA. Moving beyond just seeing them co-localized.
Exactly. It tackles those pain points of reading descriptive papers. You see things together, but you don't know if it's cause or effect. This lets you intervene.
Very cool. And the list goes on. Stress granules, telomeres, the centrosome. They really pushed the versatility.
They even used a myosin motor to drag GAPDH mRNA to the leading edge of a migrating cell.
Suggesting a role in cell movement.
Potentially, yeah. It hints that maybe cells position specific RNAs to help them crawl or navigate. Lots to explore there.
And they could watch this happening in real time and control it with the ABA dose.
Yes, the extended data figures show this beautifully. They could add ABA and watch the RNA move, vary the ABA concentration to get different amounts of localization, and then wash out ABA to reverse it.
And even track single molecules moving along microtubules.
Oh, using MS2 tagging on the RNA, you can literally see the CRISPR-TO machinery carrying the RNA along the cell's transport tracks. It gives you incredible dynamic detail.
Okay, so the tool works, it's versatile, it's controllable. Now the big payoff. What are the functional consequences of moving RNA around like this?
Yeah.
Especially in neurons, which seem like a focus.
Right. This is the really exciting part, moving into primary cortical neurons, actual brain cells. They used CRISPR-TO to move ACB mRNA, the beta-actin message, down those long neuronal projections, the neurites, right to the tips. They used a kinesin motor for this long-distance hauling.
That's quite a journey in a neuron. What happened when the active RNA got there?
So this was a key finding. They showed using clever reporters like Denver-2 photoconversion and synapse.
Ways to see where new protein is made.
Exactly. They showed that the ACTB mRNA arriving at the tip was actually translated into beta-actin protein right there, locally.
Okay. And what did that local protein do?
It boosted the formation of these dynamic little feelers called philopodia, things neurons use to explore and connect.
So, more exploration.
Yes. But interestingly, it also seemed to inhibit the regrowth of the main axon, the main output cable of the neuron.
Wow, okay. So a specific localized effect on neuron structure and growth. How else did they use this in neurons? There was a screen mentioned.
Yeah, this is super powerful. They didn't just look at ActBead. They did a screen using CRISPR-TO to systematically move a whole collection of different mRNAs to neurites.
And looked for what?
They looked for which RNAs, when forced to the neurite, actually promoted neurite outgrowth.
Like a functional genomic screen for RNA localization effects.
Precisely. And through that screen, they identified SDMN2 mRNA localization as a key driver of neurite growth, something potentially missed by just looking at overall RNA levels.
That really highlights the power of manipulation over just observation, discovering function. And they combined CRISPR-TO with other methods.
Yes, they threw the kitchen sink at it in a good way. Real-time imaging with MS2, measuring local translation with ribopyromycylation, even local proteomics to see exactly what proteins were being made nearby. A multimodal approach.
So, stepping back, what's the big innovation here? What's the core breakthrough?
I think the fundamental shift is taking spatial transcriptomics from observation-making maps to intervention, actively manipulating the spatial organization of RNA. It opens up completely new experimental avenues.
And the tool itself, CRISPR-TO.
It's designed as this versatile, programmable module. You can target different RNAs with the gRNA, different locations with the motors or adapters. They even suggest potential for light or chemical control, maybe like the DCAS9K-ARB system is for gene silencing. It could become a standard tool.
Are there limitations acknowledged in the paper? Areas for future work?
Oh, sure. They're upfront about needing to better quantify transport efficiency in different contexts and carefully check for off-target effects, especially in complex tissues. Like, are you accidentally moving other RNAs you didn't intend to? That needs careful validation.
Makes sense. But for anyone trying to understand biology, this feels like a major step towards, really testing how things work rather than just describing correlations. It helps get past some of those frustratingly dense descriptive studies.
Exactly. It lets you directly probe the causal links. Does RNA location drive development, synaptic plasticity, disease states? Now we have a way to potentially find out.
It really gets you thinking about future directions, doesn't it? Could you say, target mislocalized RNAs in neurodegenerative diseases? Try to put them back where they belong.
That's the kind of translational potential people will definitely be thinking about. If we understand the rules of RNA localization and what goes wrong in disease, maybe we can design interventions based on tools like CRISPR-TO.
So the really exciting takeaway here is this newfound power to actively sculpt the RNA landscape inside living cells. Unprecedented control.
Which leads to the question for you, our listener. Thinking about your own area of biology, what questions, maybe ones that seemed impossible before, could you now tackle with a tool like CRISPR-TO? What cellular mysteries might be unlocked by controlling RNA's address?
And a final thought to leave you with. Could this ability to precisely place RNA molecules lead to entirely new kinds of cellular engineering, or maybe even targeted therapies that work not just by changing RNA levels, but by changing RNA location? Definitely check out the paper. It's worth the read.